Okay, final video of the week. We are actually going to go into the last part of innate immunity, innate immune cells. We're not going to do all of the cells. We're just going to talk about the first part of it, but we are actually going a little bit deeper now. So far, we have already talked about barriers. Got a rule of threes here. We've got barriers, complement, we're about to do cells. For barriers, we have three of those: mechanical, chemical, microbiological. For complement, we get 2 threes, how they're activated, the alternative pathway, MLB pathway, MBL, sorry. Always want to think baseball, I guess, or the classical pathway. And once they are activated, a whole bunch of different proteins come together and trigger and do a whole bunch of different things. But in the end, it does one of 3 things, or sometimes a combination of 3 things. It kills the pathogen by forming a membrane attack complex, a MAC. It labels the pathogen for destruction by other innate immune cells, and it signals to other cells in the area that there is a problem. So we've got our first 2/3. The last thing we're going to be adding is innate immune cells, the cells of the innate immune system. And when I say talk about the innate immune cells, there's a lot of different things that we can be talking about and a lot of different cells. We are going to focus on the big 2 of innate immune cells, the neutrophils and the macrophages. There are similar things, and it's not that all of this doesn't apply to basophils, eosinophils, and mast cells as well, but there's some specificity, and we're going to stay a little bit more general as we're just learning kind of the big aspects of the innate immune system and getting into innate immune cells and what they do. And the reason that I bring this up is because just like complement can be triggered by antibodies, so can immune cells, and mast cells actually function mostly through antibodies, and it gets complicated. So what do innate immune cells do? I remember what innate cells do and kind of keep track of all of the things that are happening here by thinking that every time an innate immune cell encounters a pathogen, it needs to race. It needs to recognize that there's a problem. It needs to alert others that there is a problem. It needs to make sure that all of the others are getting to where they need to be. And then it needs to eliminate whatever that problem is. So for most of next week, we're going to talk about the alert, converging, and eliminating aspects of innate immune cells. We're just going to start with recognition. How do innate immune cells recognize that there's a problem? Well, we've already hit on this in a lot of different ways. We've already talked about how key PAMPs are. We've talked about how those PAMPs and those differences between pathogens and our cells are generally what allows the innate immune system, all aspects of it, to be activated and hurt pathogens without hurting us. We had those chemicals, lactoferrin, which only work to release iron to us, but not to pathogens. We talked about how certain characteristics of the cell membrane allow antimicrobial peptides to bind and disrupt membranes. We talked about how lysozyme binds to cell walls, which we don't have. We talked about how the lack of complement control proteins are what allows complement to be activated by the alternative pathway, and how specific patterns of sugars, the way that mannose is found on cells, will allow complement to bind through the MBL pathway. So the innate immune cells are not going to do something all that different. They too are going to be looking for PAMPs and DAMPs. And they will detect those PAMPs and DAMPs using pattern recognition receptors. These pattern recognition receptors are actually on the innate immune cell. The PAMPs are part of the pathogens. So these are the things that are actually on there, or in there. Whereas the pattern recognition receptors are actually the arms of the innate immune cells. And a single innate immune cell is going to have more than one pattern recognition receptor. A single innate immune cell is going to be able to recognize viruses and bacteria and flagellum from parasites. It is going to have basically its eyes out for all of those different things. Remember, it's not super specialized. It's not the cardiothoracic neurosurgeon. Actually, I guess you would have a neuro, it doesn't matter. It's generally looking for a problem. An EMT is going to be able to identify generally what's happening, even if it can't do a specific diagnosis and, on the spot determine that it is actually, SLE versus different forms of lupus, but innate immune cells have lots of different PRRs. It will say what kind of PAMP it is, and at least generally be able to respond in the right way. There are lots of different PRRs. There are a bunch. But we are going to focus, again, just like we only focused on complement for the main soluble, non-cellular part of the immune proteins, we are going to focus on, or at least start by talking about the most common or the most well-studied, again, toll-like receptors. There are a number of toll-like receptors that an innate immune cell will have. These toll-like receptors will be, and actually pattern recognition receptors in general, will be both on the surface of the cell looking for things outside the cell, but there will also be internal pattern recognition receptors. And this is where I also have to say, remember, we keep coming back to every cell in the body typically plays at least some role in the immune system. So a lot of our cells have, even though they're not immune cells, not just macrophages and neutrophils, have pattern recognition receptors inside them as well. Because remember, viruses can infect any cell. There are some viruses that attack immune cells. HIV specifically really likes our dendritic cells, some macrophages, and likes CD4 T cells. But a lot of viruses just attack our respiratory cells. Those aren't technically immune cells, but we want our cells to know if there's a problem. So we're going to focus on toll-like receptors and pattern recognition receptors as part of immune cells, but know that all of our cells have pattern recognition receptors, at least to some extent, so that no place in our body is safe for pathogens. So as I said, each immune cell and each cell in our body will have more than one pattern recognition receptor. And in fact, most immune cells will have pretty much all of them. And each pattern recognition receptor does recognize something different. So in this figure, you can see both where they are located, but also what each of these pattern recognition receptors, these specific types known as toll-like receptors, recognize. So you can see here that we have lipopeptides, see lipids and peptides coming together, fatty proteins. That TLR1 and 2, they actually work together, form a complex to recognize. Peptidoglycan, if you've taken microbiology, that's a key component of the bacterial cell wall and unique to bacteria. We don't have it. In fact, even fungus doesn't have it. So when TLR2 binds to peptidoglycan, it can trigger downstream signals that are specific for bacteria. And that peptidoglycan would be different than seeing, certain types of DNA. I mentioned that there are differences in the way DNA is methylated, that different methylation, the different sort of proteins it's associated with it will trigger responses. And so again, it's not that the innate immune system just says, problem, it can at least kind of identify like fire versus, intruder versus heart attack. So these toll-like receptors and pattern recognition receptors in general do have some degree of specificity, even though they are fairly general. And so the responses will be different. And we are not going to spend a lot of time talking about these signaling molecules. But, we didn't just learn about cell signaling in intro bio because it was nice to talk about. These cell signaling molecules are really important because different signals will lead to different responses. And a lot of these signaling molecules are really important targets for altering the immune system. We don't want to just turn our immune system on or off. We want to turn specific aspects of our immune system on and off, even with treatments, in order to be able to have it do what we need it to do, but not do too much. And so different downstream pathways, lots of different signals being activated, will lead to different responses. For almost all of the pattern recognition receptors, all of them will typically lead to the expression of some sort of cytokine, some sort of signal that lets others in the area know that there's a problem. Just like an ambulance or a fire truck, as you could hear outside, has flashing lights and a siren, these cytokines are the sirens. So when a toll-like receptor is triggered, there are going to be certain cytokines. And again, different triggers will lead to different responses. Here we have interferons being triggered by some of them. These are going to be interferons or cytokines that basically alert that there's a virus. Others, MyD88, which is a downstream signal, will lead to the expression of different sorts of cytokines and different, basically different responses. As I said, there are more than toll-like receptors, and not every cell in our body per se has toll-like receptors, but they do have usually some pattern recognition receptors. STING is a very common one that, of course, I want to say all cells, but then I'm sure there's some exceptions, will have. STING is looking for things that are inside cells that are trying to hide, whether that is some breakdown products of intercellular bacteria or viruses, which again can get into any cells. STING leads to generally an intracellular response. It leads to interferon as a cytokine because it's warning others, hey, you want to shut down because somebody's trying to break in. An intracellular bacteria or a virus is trying to get inside, so go on lockdown. Interferon is a great, type 1 interferon in particular, talk about interferon more later, is a great warning of that. Similarly, we have other receptors, caspase-1, ASC, and LRP3. All of these are different enzymes that, again, can do different things depending. You're going to be on the lookout for extracellular bacteria. These are going to be different than the interferons. This is going to secrete more IL-1. Again, we're going to talk more about these cytokines when we talk specifically about the alerts. But just know that one of the common triggers of all of the pattern recognition receptors does tend to be the expression of cytokines. Warns things and gets the party started, so that next week we can talk about more what it actually does now that it has recognized that there is a problem.